every breath,
Wala pa si sir. Wala pa.
every heartbeat, and every memory we carry. All of these exist because two powerful systems work silently behind the scenes. The nervous system, our body's electrical communication network, and the endocrine system, our long distance chemical messenger system. Today, our group, Jeric, JB, Amira, and Sophia, will demonstrate the laboratory procedures for these systems using clear step-by-step explanations and digital animations.
To begin our laboratory procedures, we start with the gross examinations of the human brain. Since we are not using any physical specimens, we will represent the procedures with animated models. First, we prepare the workspace to ensure safety and sterility. Before handling the brain, we first cover the dissection board with plastic to prevent contamination. Each student must wear complete PPE, gloves, mask, and lab gown to ensure personal safety.
Next, we examine the major structures of the brain to understand its overall organization. We begin examining the whole brain. The cerebrum is the largest region responsible for voluntary movement, memory, and thinking. Below, it lies the cerebellum, which coordinates balance and posture. The cerebrum has four lobes. First, we have the frontal lobe, reasoning and motor control. Second one is the parietal lobe, which is for the sensory and interpretation. For the third one, we have the temporal lobe, which is the hearing and memory. And for the last one, we have the occipital lobe for vision.
Moving deeper into surface anatomy, we now observe the sulci, gyri, and fissures. On the brain surface, the raised folds are called gyri, while the shallow grooves are sulci. The deeper ones are fissures. These structures increase surface area, allowing more neurons to fit inside the skull.
Shifting to internal views, we explore the mid-sagittal and coronal brain sections. In the mid-sagittal section, we identify the corpus callosum, which links the hemispheres. And in the brainstem, we have midbrains, pons, and medulla that controls the vital function. In the coronal section, the green matter forms the outer cortex, while the white matter lies deeper.
Proceeding to a functional test, now we demonstrate the pupillary light reflex. This test evaluates the optic nerve and the brainstem pathways. The room must be dim. A divider covers one eye while we shine a pen light toward the other from the side, not directly, to avoid startling the subject. A normal response is pupil constriction. We repeat the procedure for the opposite eye and observe how both pupils change once the light is removed.
Before triggering any response, we establish the subject's baseline vital signs. The subject sits for 2 minutes to stabilize the vital signs. We measure the pulse rate, the blood pressure, and respiratory rate. These represent baseline parasympathetic activity.
Once the baseline is recorded, we introduce a stressor to activate the sympathetic system. Answering rapid multiplication questions activates the sympathetic nervous system. Pulse, blood pressure, and respiratory rate increase as the body prepares for action.
After the stress phase, we guide the subject through relaxation breathing. Afterward, the subject performs deep breathing for 10 minutes. This activates the parasympathetic system, decreasing vital signs and restoring equilibrium.
Transitioning to the endocrine system, we begin with a brief overview of hormonal control. The endocrine system controls the body through hormones, chemical messengers that travel in the blood and bind to receptors, triggering cellular responses. Negative feedback loops prevent hormonal imbalance and maintain homeostasis.
To begin the experiment, using a microscope, place the prepared slides and observe under LPO. Then shift to HPO for confirmation of structure.
First, we look at the ovary. Located in the pelvic cavity, the ovary produces estrogen and progesterone. Under the microscope, follicles at different stages are visible, containing oocytes surrounded by granulose cells.
Next, we move to the thymus. The thymus in the mediastinum matures T lymphocytes. The cortex appears darker due to dense T cells, while the medulla contains lighter Hassel's corpuscles.
Following that, we examine the adrenal gland. The adrenal gland sits above the kidney. Its cortex has three zones: glomerulosa, fasciculata, and reticularis, while the medulla secretes epinephrine.
Then proceed to the thyroid gland. Its follicles store colloid surrounded by simple cuboidal epithelium that produces T3 and T4.
Finally, we end with the testes. The testes produces testosterone and sperm. Under the microscope, seminiferous tubules contains developing sperm cells.
And that concludes our laboratory demonstration for the nervous and endocrine systems. We explored the major structures of the brain, the pupillary light reflex, and how the body responds to the sympathetic and parasympathetic systems. We also examined the endocrine glands under the microscope and identified their key hormones and functions. Through these activities, we learned how these two systems work together to keep the body balanced and functioning. Thank you for watching and have a nice day.